Good morning, one and all. I cordially welcome all the participants. Resource person, today's resource person, Mr. T. Praveen Kumar, Senior Vice President, Bio for India Limited. And on behalf of the management, principal and staff of Vigyan Pharmacy College, I cordially welcome all the participants to the Day 4 FDP program by Vigyan Pharmacy College in association with National Research and Development Corporation. Now, I request our Honorable Principal Sir, Mr. Srinivas Babu Garu, to speak a few words on this event.
Thank you, Vishwanath. So, today's session on the eve of e-faculty development program on behalf of Vigyan Pharmacy College. We welcome my dear student, Praveen Kumar, Senior Vice President, Bio4, on the topic product development in lieu of addressing challenges during technology transfer. So I thank all participants for the last since 3 days on the various topics. I hope we are enlightening all the information to the various pharmacy fraternity and different multi fraternity related to patents and all. So I wish all the best. Thank you. Thank you, Anand.
Thank you, sir. Now I request Ms. Geetika, Assistant Professor of Vigyan Pharmacy College, to read out the profile of Mr. T. Praveen Kumar Garu. Geetika, please.
Thank you, sir. Good morning, everyone. It is my pleasure to present you the profile of our speaker, Mr. T. Praveen Kumar, Senior Vice President, Bio for India. Mr. Praveen Kumar obtained his Master's in Pharmacy in 2005 in KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. Mr. Kumar was trained in Cipra Labs Limited from November 2005 to October 2006. He worked at Orchid Healthcare Limited, Chennai from October 2006 to April 2009. He worked as Senior Executive at Hetero Drugs Limited, Hyderabad from April 2009 to September 2009. He worked at Recon Pharma India Private Limited, Hyderabad from September 2009 to July 2015. Mr. Praveen Kumar successfully faced different customer audits and its compliance, actively participated in US FDA audits for general GMP products audits and the API manufacturing site. Sir, I welcome you to this EFTP program.
Yeah. Thanks for presenting the profile. So it's really honor for me also to present my thought process because basically I'm a formulation development scientist who is having around 15 years of experience in the field of pharmaceutical development, where we have been developing a lot of molecules for the US FDA as well as the other regulatory markets. So across my career, we had faced a lot of challenges during the product development and also during the technology transfer. So I would like to share my thoughts on this topic of product development in view of addressing the challenges during technology transfer. I just want to make a note that there is one small correction in my designation. It's the Associate Vice President. It's not the Senior Vice President, but that's okay. That's fine. And so I'm really privileged to participate in this program on the faculty development program. and for the Institute of Ignorant Pharmacy College in association with National Research Development Corporation. So coming to the details of this presentation and outline of this program, I would like to emphasize on a certain topics, like before entering into details. First, I would like to take a few minutes of time, even though we know everything about what is formulation development and how do you formulate a drug, but just I want to give my insights into that. So I have selected a brief outline of this topic as what is formulation development and why do you formulate a drug substance, and what is the role of product development in various stages throughout the drug development process, and how do we understand the product development through a quality by design to understand the challenges of the technology transfer. And next, coming into details of what is a technology transfer and what are the process and scale up understandings for the technology transfer, and a few examples with respect to the unit operations, what we have, even though they look simple, but yes, there is a lot of things that we need to consider during the development part to understand how exactly we can do a technology transfer without any issues and without any challenges, and the overall conclusion of this. So coming into what is a formulation development. Basically, a formulation development is a key area of the product development that can determine the patentability, life cycle, and ultimately the success of the pharmaceutical product. So, and it's an art of science that transforms a powder substances like the APIs and the ingredients into a quality drug product in a specific dosage form by developing an appropriate formula process to attain the therapeutic equivalency. So this is what is a brief of the formulation development. We take the input materials, we convert it into a specific dosage forms. It might be a solution, it might be a tablet, it might be a capsule, it might be an injection. But there is a lot of science where we need to consider in order to have a transformation from that powder to a specific dosage form. That science of converting it is we treat it as a formulation development or the product development. Coming to why do we formulate a drug substance? Why not we take it internally as such as a powder or internally as such material? Because the basic requirements for formulating of a drug substance would be a clinical need. As such, the powder would not be a surrogate to have a clinical efficiency. It has to, because the basic requirement is that it's a BC, like you have to have a solubility, then you have to have a permeability, wherein all the powders or all the materials doesn't have that capability to meet the clinical needs. And the second thing was to meet the patient needs, because the patient himself they may not be able to take up the powders as such in order to have an effective absorption system. And the third was the industrial needs, like even the packaging, considering the development part, packaging part, the other parts, industry has some challenges in handling those kind of products. So it's always easy to handle a specific dosage form rather than a specific material. So that's where the industry needs comes from. And the final part was the targeting the delivery of the drug substance. As such, some of the drug substance needs to be targeted to the specific sites, or some of the drug substance needs to be absorbed at the specific site. Or when you consider an example of proton pump inhibitors, they might have an acid degradation. So the API needs to be processed to address those challenges. So for that, we have to create a dosage form that goes into the solution, it disintegrates, goes into the solution. Once it gets into the solution, that it will be absorbed in the systemic circulation. They might have a liver metabolism, gut wall metabolism, or all sorts of things, but the absorption would be enhanced while formulating it as a drug product. So that's the reason why we need to there are unmet needs to formulate a drug substance. And coming into the role of product development, so how a product development is being involved in the various stages of drug product. We can see the development stage of the API to the formulation. We have a lot of stages involved here. We have a development screening, we have an optimization, we have a preclinical stage, we have a phase 1 clinical trials, phase 2 clinical trials, phase 3 clinical trials. Post the phase 3 clinical trials, we have a commercialization segment here. So once you come till the preclinical stage, it covers under the drug discovery. So drug discovery covers with respect to the what is your pre-formulation studies, what are the formulation support for preclinical tests, and how do you develop a dosage form, what is the basic criteria for development of a dosage form. All these will be covered in the drug discovery part. And coming to the clinical development, once a dosage form is developed, there are unmet challenges there. How do you have a formula in place? Like how do you have the robustness that you need to build into the formula? That covers your formulation development with respect to the formulation optimizations, and what are the process that we need to follow. You need to follow, if it comes to a tablet, you need to follow a bed granulation technique, direct compression technique. So what is the process that have, and what are the critical parameters that are being there here to cover those to cover those processes. And once you have a formulation in place, once you have a process in place, then comes your clinical supply that goes out for doing a phase 2 or phase 3 clinical studies. And once that clinical studies passes, then the real challenge starts in the life cycle management. The life cycle management demands for the tech transfer of the scalability of the formulation from your lab scale to your commercial scale. And that in some cases may demand for a reformulation, may demand for an expansion, or might demand for a variation in, even like you can go from an RX to the OTC switch. Like you can even change the dosage form, change the indication. So that's the challenge that goes into the life cycle management during the commercialization. That's the reason why we have to consider the technology transfer aspect during the formulation development and the process development to address these kind of significant challenges in the life cycle management. So what would be the various stages of the formulation development or the product development? The various stages includes your pre-formulation, prototype development, scale-up and technology transfer, and the commercialization. So even though it looks like 4 stages, we can. I personally feel it has to be segregated. It's only into 2 stages. One is your pre-formulation and the prototype, and from scale up to commercialize, it's a single activity that is the technology transfer. So we'll go into in-depth with respect to each of the stages and how do we correlate it back to the technology transfer. What kind of challenges would you would anticipate from the technology transfer perspective for pre-formulation and the prototype development. So coming to the pre-formulation stage. So in the pre-formulation stage, we try to understand better about the drug substance, better about the individual ingredients, their solubility profiles, stability profiles. And then we cover this into a sphere of pre-formulations with respect to the relation on the development challenges. So once you consider, suppose you take a drug substance can be categorized into a class 1, class 2, class 3, and class 4 as per the BCS classification system. That means it has the high solubility or a high permeability, or it has low solubility or a high permeability as per a class 2, or even though if it has a high solubility, it has a low permeability, and the class 4 is it doesn't have a solubility and permeability. This is a basic understanding what we take upon on the drug substance part. But when you really go in depth in understanding how the drug substance plays a role with respect to your technology transfer, there are a lot of other segments, like you need to understand your crystal structure, you need to understand your particle size distribution. You need to understand your morphology. You need to understand how a drug substance is going to play a role in just creating or in anticipating the challenge of a drug technology transfer. For example, we suppose you have a BCS class 1 drug, which is having a high solubility and a high permeability. Then we personally feel, as a development scientist, we feel that, oh, we have a very good drug substance, it's highly soluble, so it may be a rate limiting step, it may not be a limiting step. Once it goes into the body also, it gets absorbed. But when you are really converting it into a dosage form, your crystalline structure of your API is going to play a role, and your solubility nature of the API is going to play a role there. Because once you go to a highly soluble drug, so the drug itself is highly soluble, there is no challenges, there is no unmet demands, and there would be no stability challenges as well. But the real challenge comes in your process. What is the type of granulation you have to select? Or you have to select a direct compression process, or you have to select a granulation process. What kind of granulation? When it's a wet granulation process, or whether it's a whether it's a roll compaction process or a dry granulation process. So what is that you have to select? And when it goes to the wet granulation, what kind of solvents you need to select? You need to select a solvent, you need to select either you need to select an aqueous solvent, or you need to select a non-aqueous solvent. So based on our development experience, we faced a lot of challenges in understanding the drug substance behavior, in understanding a drug substance crystalline structure, in understanding the PSD. For example, we had one product which is having a BCS class 1 drug, so there is no issue with respect to the dissolution. We have taken a lot of, even we can manufacture with high particle size in lieu of dissolution and all. But the challenge we encountered is on the compressibility of the granules. The moment you have more particle size and the more crystalline nature in the API, your API is not going to get compressed into your compression cycle, irrespective of the various concentrations or the formulation demands you have, or the formulation ingredients you have. You will have a lot of challenges. So considering those kind of challenges, you have to anticipate in your pre-formulation, and then you have to take those challenges in consideration while doing a pre-formulation. What is the particle size I need? What is the crystalline morphology I need? I need a larger crystals, I need a smaller crystals. So those kind of things needs to be considered while developing the product, by in view of anticipating your challenges on the technology transfer. These kind of all challenges put together forms a smear, in that you have the API characteristics, you have the excipient characteristics, how do you convert, what kind of granulation you need, what kind of studies you need, when I need to use a granulation, whether I will have a better solubility profile, better stability profile, do it impact on my stability. All these things has to be considered, has to be put in place. It has to cover in the ideology of the pre-formulation, and then we have to go ahead with the formulation prototype development. Yes, Lakshmi, anything you want to have? Hello? Hello?
Sir, we can hear you, sir.
No, I think I have one participant by the name Lakshmi Devi who has raised her hand. Can you just put her on speaker so that we will clarify her doubt and proceed further?
There is no person who lifted hand, sir.
No?
No person, sir.
Okay, that's
I've
fine.
checked it. But anyhow, we will see that question and answer section at the last, sir.
Okay, fine. That's fine.
So 1 second, sir. Let me announce it. I request all the participants to ask their questions at the end of the session. Thank you. Sir, you can please continue.
Yes. So this is how what we have to consider in the pre-formulation stage. Then the second stage of development goes into your prototype development stage. This prototype development, it all deals with your how you formulate your drug product. What are the various ingredients you are going to select? Suppose it's a tablet or a capsule, what kind of binders you are going to select, what kind of disintegrants you are going to select, what kind of lubricants you are going to select. So all these things needs to be captured during the ideation stage and conceptualization stage. You need to put in all the concepts in place. You need to anticipate the challenges in place with respect to the individual unit operations. If I use a binder solution, what kind of binder I need to select, what kind of profile I am anticipating. All these things goes into the idealization and the conceptualization stage for the prototype development. And then we will have an experimental design by putting quality target product in place. So how this covers is, it covers into the risk management. So you have a slide here on the right side where the process monitoring and the continuous improvement is our target. And that target needs to be attained by creating a design space. And that design space will be defined, in the design space will be defining the control strategies and the product characteristics and the verification. And that needs to be built in from the quality target product profile. So basically we start our development here with you by defining a quality target profile, by creating a proper ideation and conceptualization stage, where in the product quality target we will clearly define what are our quality attributes. What is that we are looking at with respect to the quality? It may be the assay, it may be the dissolution, it may be the RS, it may be the stability of the product, it may be the bioavailability of your drug. So all these things covers your quality, critical quality attributes of your drug product, and what are the processes we are going to put in place for attaining these quality attributes. Like what are the processes, what are the critical process selections we are going to make for each process selection, how critically we are evaluating the process parameters. So all these things comes in your idealization, and it has to be part of quality product profile by targeting your technology transfer as your final stage. And then subsequently, not only the formulation, we have the analytical control strategies also. Like during analytical, even in analytical also, we see a lot of challenges during the transfer of the method, during the running of the method with respect to the column, with respect to the peak shapes. All these things will be covered. So as a cumulative, if you talk on a product development, during ideation and conceptualization, the moment you go to the conceptualization, you should have a clear on the process. What are my quality attributes? What are my process attributes? What are my analytical attributes? And how am I relating these attributes to the technology transfer? Or how am I converting these attributes to make a product commercially available to the market without any challenges or without any recalls? I would call a product recalls, because the moment your product is not performing in the market, it doesn't go well with the patients. Patients will have a lot of complaints. You will have a lot of recalls. So considering all these scenarios, we have to create a quality target profile. Once you create a product target profile, then your development work starts by designing your experiments for attaining your quality target profile. How do you design your experiments? Whether you design one-on-one experiment, which is the most primitive experiment, 1 factorial design, or create a design for of experiments to understand how your product is behaving, what process variable is going to impact your development prospect, or what formulation is going to impact your formulation variable is going to impact your quality attributes. and what process variables is going to impact your process parameters. So all these things will be studied in the experimental design part, and we create a design space by optimizing the formulation and by optimizing the process, and then propose a control strategy during the product development before the scale-up batch. That control strategy has to be taken further through a process characterization and a verification on the commercial scale, and then it goes into, once the commercialization done, then you will have a continuous process monitoring, and in case the process improvement is required, we can take a process improvement. But all these factors needs to be considered before you start your development program, and then that creates your robust development, robust process, and there would not be any unmet challenges in your technology transfers. So this is how we have to take into consideration for the prototype development. So then once during your prototype development, once you have your ideation, concepts, everything, so how do you create your ideation concepts with respect to a quality by design? How do you put your product quality first and then design your drug product to have a product that is very effective and it doesn't have any challenges on your technology transfer? So for this, we need to understand the product development through a quality by design. In the quality by design, on your right side, we can see a target product profile, which we talked about. And through this product profile, we'll have an early product development which relates to the pre-formulation of the API, with related to the pre-formulation of your excipients, all those things. And at this stage of quality target profile, we have to have a risk assessment done. So what kind of risk I'm anticipating? What kind of challenges I would face for the formulation? So those risk assessments needs to be done. The best tool for doing a risk assessment is your FMEA, failure effective mode of analysis. And using that, you can identify what are the various risks and what are the approaches we need to follow throughout the development process to factor this risk during your quality target profile. Then we design a process, and once a process is designed by the design of experiments, then based on the understanding of the process design, we have a control strategy. During designing a control strategy also, we have to have a risk assessment in place. Are my process variables or my formulation variables are sufficient, or the study that I have done is sufficient to ensure that my process will be consistently delivering the desired quality of the product. So that risk assessment for each and every stage has to be put in place and has to be considered. And based on that risk assessment, we can go ahead with identifying the probable variables with respect to individual unit operations, work on the technology transfer, and mitigate those process risk, by identifying what are the critical variables for each unit operation and putting a conformance or manufacturing or a process qualification in place by taking into the variables here and monitoring it through a continuous process verification. So at 4 critical stages, we have to understand the product risk assessment. One, it is at the QTP stage, one it is at the control strategy stage, one at the process risk assessment stage, and one at the continuous process verification stage. Once you understand this and put this part of your development program, I think that's where we can address most of the challenges during the technology transfer. So how do you understand this? So we have a lot of terminologies, quality, design, and all. How do you understand this? We will understand this quality by design only by reducing the process risk, product risk, and reducing the product risk and increasing the process knowledge throughout the product development and manufacturing. And there is a stage where your product risk and your process knowledge will bisect each other. This stage is what we call as a technology transfer stage. Once a technology transfer is done, then we will have more understanding how the process is behaving on the commercialization stage. That creates a more betterment on the process knowledge. And the more you have the process knowledge, the more you can reduce your product risk and the more you can reduce your customer complaint. And this bisection stage is the stage where you critically consider that stage as your technology transfer, and where you can put your design space, all control strategies in place. And this is the stage, the green stage is what we call as a continuous improvement, and where your process will be monitored, improved after the technology transfer. And this is the stage where the first initial stage is the red zone, where you understand your product, understand your product process, and all these kind of stuff, where you will have a high risk zone where you cannot have a better product without understanding your product and process. And the moment you have more process knowledge, your product risk will go down. And this is where your continuous improvement takes in place. And how do you do that? How do you improve your process knowledge? How do you reduce your product risk? So this can be done through a process development, where the major objective of your process development is to propose a control strategy by effectively understanding the risk associated with the individual unit operations, and then defining those critical process parameters in lieu correlated with your quality attributes, and that acts as an essential guide to your development process. And how you understand better, how do you have a quick process, like how do you increase your process knowledge? This is something that you will have a multiple feedbacks mode on your QBD, where you define your QBD quality. Suppose you define a quality target profile, you have to go back to your early product development and see that whether this product quality profile is being covered in the earlier product development, or what kind of variables need to be additionally put in place. That's the same way you have a process development. Then this process, based on your understanding of the process development, you have to go back and redefine your quality target product profile to have an effective understanding or reducing the risk of your product. And the more critical would be your technology transfer and the continual manufacturing, where during the technology transfer, once your product risk assessment or process risk assessment is done, it goes to the control strategy. However, if you don't have a better control strategy in your development program to understand the process variables and the quality variables, this always demands for a process development at the technologies transfer. You can see an arrow going back to the process development during a technology transfer. Why this happens is because you have not understood your process. You are not spent sufficient time in understanding your product. What are the risks associated? How do I improve my process? What are the variables in it? So if you are not doing that, we will have an unmet demand in technology transfer to go back and look into your process and redevelop your process. Even though on a smaller scale, if you are having a very minimal challenges here, but during your continuous process verification on a commercialization scale, you will have a bigger risk in going back to do a process development that demands for a SUPAC level on a regulatory front, the scale up and post approval challenges, how do you justify and all. So to reduce this bigger arrows and the smaller arrows going back to your process development, we have to have a control strategy well defined, well established before you go to a technology transfer. So once you say a technology transfer, so you have put your development thought process, everything in place. Then what is technology transfer? So the scale up or a technology transfer is the body of knowledge available for that specific product and process. That includes your critical quality product attributes and the process attributes, process parameters that has a capability for manufacturing and processing a control technologies to have an effective control systems in place. This is what we define it as a scale-up or technology transfer. So how do you define this? So we define it through a product understanding. We will define it through a product, in product understanding, we have the criticality for the process. We have the criticality for the excipients. We will have a CQAs that are to be considered for the control strategy. And we need to identify the attributes to mitigate these control strategies. Then once you have this stage done in the product understanding, then it goes to a process understanding. This is where we have to spend more time in understanding your unit operation, in understanding your operating parameters, in understanding the behavior of your raw materials, and in identifying the risks that are being associated for the technology transfer. And accordingly, we will have an unit operation design space that characterizes the process variables, the process capability, and defines a control strategy assessment. Once you have this assessment done in your process identification and the technology transfer, then it goes to your control strategy for the manufacturing of the larger batches, where you have a control strategy elements and you have a control process verification in place, covering your process controls and the testing parameters. Once you cross this stage of process understanding, you have a better process understanding, then you anticipate a less challenges on your control strategies and the continuous process verification. Otherwise, you will always revolve around your process. How do I put my process to be effective? You will do one trial and then go back and say that no, this trial is not working. So all these things has to be considered in place to improve your knowledge on the product as well as the process. So how do you improve your product knowledge? During product development, we have to create a knowledge. It's nothing you are going to get somewhere else about that process, but you have to create a knowledge on your drug substance, on your variables, on your drug delivery, analytical methods, package, everything. This needs to be a part of your development program. You have to create a knowledge. And then based on the knowledge that is being created, you have to identify a risk, identify the process variables, identify the process parameters, and then you have to prepare to execute with sufficient details and controls on your development batch, and then go to a scale-up and validation stage. And if you can see a graph that goes to the development and process monitoring from here to the here, you will have a stage. This is where we call it as a cross-sectional phase, where you have more process or an equal process understanding and the product understanding. The moment you enter into creating more design space, more identifying more variables, your process will become more robust before your commercialization. And this is where you have a flattened curve. And this stage needs to be verified during technology transfer and identify with respect to the process variables and propose a control strategy. And therefore, you will have a seamless execution on the continuous improvement. So in your product development, you have to create a knowledge, technology transfer. You have to enhance your knowledge by putting controls in place, by executing various process controls and having a better understanding on the process capabilities. And then you manufacture, execute for the continuous improvement. So considering this stage, how do we execute? What are the controls we need to bring in? So we have a certain goals for technology transfer. What should be our goals on the technology transfer is that we need to establish a successful commercial manufacturing for the drug product by establishing a robust manufacturing process. So the term robust itself indicates that you should not have any issues, any challenges here with respect to the manufacturing process. So you have to create or establish a successful manufacturing with a robust manufacturing process. And the second main goal is to gather all the knowledge about the product behavior and put a control strategy in place with respect to the raw materials, with respect to the process, with respect to the formulation. And the third one is to transmit this product knowledge and process knowledge to the manufacturing side or to the manufacturing team to understand the product development and to understand the unmet challenges that they are going to have it in the technology transfer activity or in the continuous manufacturing. The moment you are not going to transmit this knowledge, then the team that goes into the commercialization will have a lot of challenges, will have a lot of issues during commercialization. And the fourth one is to ensure that the variabilities of the process parameters are controlled and that are sufficient for manufacturing the drug product. It's not only you have to define whether it's a control, but you have to also define whether they are sufficient or there are any additional things needs to be put in place for having a better control. And the last one would be the establishing the process parameters through a risk assessment by adapting various tools during development and determining the design specs for effective manufacturing. So why we need to consider all those goals in your, all those variables in your product development and the goals in your technology transfer? Because you have to have a continuous manufacturing. The moment you are not covering that, it leads to a product discontinuation. So all these goals, management system, knowledge management, quality risk management, any corrective actions, any controls need to be put in place is all covered by ICH Q10 pharmaceutical quality system, which talks on the development phase to a product continuous life cycle management. Now, what happens if your process knowledge is not sufficient to understand your drug product? So, any insufficient process knowledge results in a poorly scaled up process. You will be in a phase where you don't even understand what is happening, what would be the variables, what is going to happen, or what would be the challenges in your technology transfer. So you should have a better understanding on your process with respect to the robust process. And the moment you have insufficient process, you will, the moment you have insufficient process knowledge, you will not have any process robustness. Your production output will be reduced. You will have a decreased process reliability, more challenges, more customer complaints, and we will not be able to handle the variabilities in your raw materials, in your APIs, in your process, in your controls, in your operations. So it's like you don't go nowhere. You will revolve around your process development stage itself if you don't have a better process understanding. And if you don't have a sufficient information, that insufficient information leads to more variations in your process and commercial stage, and that increase your number of defects and issues. So how do you understand better? How do you have a process and scale up understanding? So this process and scale up understanding will be understood through models. So one model that is widely followed is an empirical model. The second one is a semi-empirical model, and third one is an mechanistic model. Empirical model talks upon your process variables, product variables, and how do you scale up your factors, with respect to individual unit models. A semi-empirical model will talks upon more into your design part or design space, control space, and all. And the last one, mechanistical model, will talks upon your process parameters, process variables, and all in your product development. Just give me 1 minute. So this is how we have your scale up and process understanding through various models for having an effective manufacturing. Now, what are the points that you need to consider during your development itself, keeping end in mind? I would like to explain this by taking few examples. One example, I'll go in detail with respect to unit operation of blending, and I'll cover unmet challenges in the process for the granular fluid bed drying and compression, just to understand, because without examples we'll not understand what we are talking. It will be more theoretical concept, so we cannot understand that. So I want to give you an understanding of the consideration, what we need to consideration on the scale up through unit operations. First unit operation, we will go through a blending. So what is blending? Just it's a simple process, right? We put up all the ingredients, we just mix it, and we take out the material and say that, oh, my process, I have a process which is having a better control. It's a very simple process, but we have a lot of failures during your scale up of unit operations with respect to your variability of your drug substance or distribution of your drug substance. Even though your blend passes, there will be a lot of issues with respect to the flow during compression that may lead to variations in your uniformity of the dosage. So even though the process is simple, but still there are a lot of failures that happens with a simple unit operations on a scale up. So once there is a failure, then where do the process went wrong? This is a simple process. Why we have to evaluate why it went wrong. So where is my process? Whether it went wrong in my variations in the symmetry, it went wrong in my variations in my product loading, or it went wrong in my time of rotation, filling level, or a rotational speed. Where it went wrong exactly, we need to identify where it has gone wrong, where it will go wrong. Because once you go to the shaft floor, comes back, then it's going to go back to your process. So before designing your process, we have to think about the failures and put your thought process in place by saying that where my process will go wrong or will yield a non-conformity of the dosage form. Just look at these two regions. It's a simple blender and you have the materials in place. So where the failures can happen. The failures can happen either in this region, or the failures can happen also in this region. So how do you address these failures when it happens here or when it happens in the different regions of this unit operations? Then is this pre-identification of the issue is possible? Yes, it is possible when you have a process understandings during your development. So how it will be possible? So you need to do a risk assessment using FMEA to identify your source of variation, and then do a proper design for the unit operations, and based on the risk assessment you can have a control strategy in place. You can optimize the risk and you can put a control strategy in place with the predefined parameters, and you have by using a established dimensional variables. For example, in a blending, we will have an established dimension variables called fraud number, where we establish the fraud number at the laboratory scale and the manufacturing scale, and we will establish an equivalency with respect to the lab scale to the manufacturing. But to do that, first we need to have an optimization that needs to be performed and define a control strategy. So one example would be like a simple example where we have done for some of the products is like a blending stage where you define your design with respect to the number of rotations and a blender occupancy, because these are the two critical variables that goes wrong. Like blend number of rotations, you have different various rotations from the lowest speed to the highest speed, considering your manufacturing equipment. And the blend occupancy is the one that goes like your blend occupancy of 30%, 50%, and the 70% throughout your operational ranges where the equipment is having the qualification ranges. You have to put these parameters at different levels. You have to design an experiment to improve your process knowledge. And then you have to generate a data. So on the blend uniformity against each of these variables at the highest and the lowest level, this comes through a 2-factorial design with a control strategy, with a control space in place. And we will generate the data with respect to the blend uniformity. And you can see by seeing these values itself, you can see that there is a process that gives you more variations at your lower speed and the lower occupancy. So this design will be the basis for understanding your process variables, and also what is that variable that might create you a variation. But in this example, we have the variable that has a lower blender occupancy and the lower rotation that gives you a more variations here. And subsequently, when you go increase your occupancies, or when you increase your speed at the lower occupancy, you will not have that kind of huge variations in your blend uniformity. So this is how we identify a critical process parameter, identify it through a risk assessment. And then you evaluate. So this process is my evaluation curve. We can evaluate through various modules, like we can create a half normal plot and see that what are those process parameters. There is an interaction with your number of rotations and blender occupancy. This is the line that the normal plot line, where the more the distance away from this line will indicate that this variable is going to create a more impact on your process. And if you see this here, when you put an interaction between this AB interaction, because this is the interaction where it's going to have more process variable, the interaction will have a higher like a transcription here, where it says that on the uniform, like on the occupancy part and the number of rotations part, So this will have more variations when you increase your rotations, or this curve will go from the lower occupancy part to over here. So this will have an interaction here, and that defines your ranges. And to understand this interaction, we do a pairwise comparison in the design experiment to understand which pair is giving you a variations, or what is that design space. Here you have a variation of 30% B to 70% B, and where you have the A factor, which is number of rotations, A minus, B minus, A plus, B minus, and B plus, B plus. They will say that it's not statistically different. But however, when you say A minus and B minus, you can have a values ranging from 2 to 2.3. And the moment you go on a variations here, your values will be reduced. And the moment you have more A plus and B minus, you will have a more variations up to 5% variation. So by understanding each and every process parameter doing these pair curves, we will have a better understanding on the process. And by plotting a control chart here, we can define our control strategy. See, when I'm going from this process from 30, 45 and increasing my process, so my uniformity is getting increased here at this process level. So I can select a variable on my scale up by maintaining that at this stage, where you will have more variations, less variations from below 1.2%. From here at 90 RPM to 60% occupancy, when I have this design space here, my variations are reduced. This is how you have to create a design space, saying that I need to use an occupancy of around 60%. And my blend variations with 90 rotations in place to have an effective control on my manufacturing for not resulting in a failure in my continuous manufacturing. So this is how we understand, we have create a design space, create a control strategy in place. So this is with respect to the blending operation. So I want to give a quick insight into the other manufacturing process also. This is a fluid bed drying process which I have selected. Like the blending process, this also have a lot of failures with respect to the scale up and unit operations. Why these failures are occurring is that there might be a process related variables here, there might be an equipment related variables here, there might be a process variables here. So is this identification possible? Yes, during development it is possible. With respect to formulation related variables, we have to understand the critical properties of the process, primary materials, what happens when it goes into drying, whether there is an improving in the elasticity, whether there is an variation, there could be a variation in my crystal structure, or whether there is a variation in the formation of agglomerates. So all these process like primary material properties needs to be considered when you are designing your process. And whether it's a low drug content or a high drug content, that how I'm defining my formulation is also critical to understand here. Or what is the solvent, binder solvent I'm selecting, low, it's an aqueous solvent or a non-aqueous solvent. What is the concentration of the solvent that we are selecting? All these variables is just an example, but the moment you do more risk assessments, more dig deeper into each and individual variable, you will have more number of questions and you will have more process and product understanding, where we can mitigate it only in the development stage. And what would be some equipment variables with respect to fluid bed drying? There would be a design, you should have a design similarity with respect to the lab scale and the commercial scale. You should have the same area distribution to avoid these kind of variabilities in your drawing. You should have a understanding on the pressure drop with respect to how the pressure is getting dropped, what is your outlet temperature, how your material flow is happening, how your shaker cycle mechanism is happening. So all these process equipment related, you should not even consider I'm done with my formulation, I'm done with my process, but you should also put your equipment variables in place. What that gives me a wrong indication, wrong variations for making my product fail on the commercial scale. That need to be put in place. And the last one would be your process related variables. Like for drying, we have to consider the inlet air temperature, fluidization air velocity, exhaust air temperature, what is my bowl capacity. All these things, when put in place, will when it is identified during your development stage, and when these parameters are covered into the control strategy, the identification of this issue is possible. And that will be done during an optimization and control strategy with establishing a process variables. For example, for the drying, you will have a process variables of calculation of your air velocity or the pressure of the proper distribution plate, etc. So once you do a risk assessment, identify a process variable, convert that risk from a higher risk zone to lower risk zone by conducting various optimizations, you will come around your process variables, or you can create a control strategy for your process variables, and that control strategy needs to be monitored throughout your process. And the last one I would like to cover is the unit operation of compression. This compression is also well known, and we'll have a lot of challenges. You'll have a lot of issues. We have gone through a lot of literature information on identifying the compression challenges, your capping, lamination, delamination. So all sorts of things, and we have certain from the formulation perspective, you have certain mitigation strategies in place for addressing each of the challenges. Like suppose you have a capping, you say that we can increase the binder, we can reduce the fines, all those things. But these challenges needs to be put in place before you do a product development. And the one best tool that we can do is an Hackett plot, where during the product development, for gaining the process knowledge, you can understand the how your process is behaving by creating a plot between your porosity and the upper branch penetration in your compression machine. You can see that the moment you are having a fill densification, then it goes to your compression. Then in the compression, you have a phase 1 and phase 2 compression, and once your tablet is compressed and it's ejected, it goes into the decompression phase, and then that decompression phase will be retained and it comes back to a field densification. So when you understand your process with respect to your phase 1, phase 2 of your compression and phase 1 and phase 2 of decompression, then you will know what process variable or what product variable, what formulation variable is going to create an impact on my scale. So a few examples of the formulation related variables is the elasticity of the granules, compressibility of the band, nature of the manufacturing, my particle size distribution of the band, what is my bulk density, what is my tab density, what is my powder flow. All these things needs to be discussed upfront, considering your technology transfer. And coming to the process variable, it's going to be the speed of compression, punch penetration, compression pressure. While defining your designing your product development itself, these process parameters needs to be controlled, and so that your issues are identified and you can put some process controls like compression rate estimation, how you can compress a rate, what is my dwell time, what is my speed, what is my powder compressibility parameters, what is the particle size I need to look into, that range, control strategy, at particular mesh. I consider a 60 mesh, how much of the 60 mesh I can have a variations. It's not like you should not put your not more than 50%, I'm fine. No, we have to tighten that with respect to the controls space, saying that I have an issue at 60% of my C retains, I'll have a range by studying the various optimizations. So once you study this and increase your process, then you will have a sound scientific principles based approach that covers your development and that creates a robust manufacturing and a safest product. And the various approaches that we are using in the development has to be elaborated in order to ensure that the product, there is a robust technology transfer for commercialization. And once you gain all these knowledge, we will be developing a robust and continually improved process. And we should not always underestimate the complexities during the scale up, and we should use all the types of tools, risk mitigation, risk identification, risk mitigation, and we should put a control strategy in order to understand the process on the lab scale and convert this into a commercial scale. That's all what I have from my side on this topic.
Thank you, sir. Thank you, Mr. T. Praveen Garu, for giving a wonderful topic on technology transfer. So I request participants to post their queries or raise their hands. We have one question from participant who is watching on YouTube. He is
Okay.
Dr. LD Patel from Gujarat. He is asking, what are the main causes of lot of failures during scale-up of unit operations?
See, the main cause of the failures would be we are not identifying, or we are not, just a minute, I'll go back. This is your major failure. We are underestimating the complexities during our development, and we are not even understanding what are the process variables that are going to create an impact, or what are the process formulation variables that is going to create an impact. Then we will have a lot of failures in the scale-up process. Once you have a better understanding, you identify these are my processes. You should be very clear during your development. These are my formulation variables that is going to impact my process. And this is my control strategy within which we have these targets in place. Then your process failures will be minimized.
Thank you. Another participant, Deba Prasad Ghosh. Please unmute. Mr. Deba Prasad Ghosh.
Yes, sir. Can you hear me?
Yeah, we can hear you. You can ask your question.
So my question is, sir, during marketing, if certain formulation related problem occurs, then what the company will do? Because during my company time, I saw that in a market, suppose metformin tablet was there, but the tablet was made with such polymers, sustainable polymers, that which tool some ghost tablets are coming out. Okay. So the patients were complaining that if the tablet is not dissolved, then how can the drug is coming out? So there was a questions from the doctor's side itself. There is similar problems appear that we need to change the formulation after post marketing. Then will the company will change the formulation, or because it will take some more time?
No, see, once your product is commercialized, And then you have a lot of complaints with respect to your product. And like, for example, your case, like I would like to elaborate that example of your metformin. So you have a metformin that is being designed during your development that has a particular disintegration time. The moment you scale up your formulation, or the scientist who is who has done a scale up on this formulation, once he was not evaluating that properly, like how is my process parameters or my formulation is going to play a role on the disintegration, then we'll have a lot of failures like that. Once you have a failure, yes, we have certain regulations in place, like we can operate around your post-approval changes. You can do a variations, like you can target plus minus. There is a guideline for this from the US FDA talking about the process variations and all, which you call as a SUPAC guidelines, scale up and post approvals. And we need to generate the data for that and submit it so that we'll have a effective controls.
Thank you, sir. And the next question is from Amartya Day. Mr. Amartya Day, please unmute yourself.
Hello.
Yeah, we can hear you.
Hello.
We can hear you. Please.
My question is, which is the best methodology to analyze or to get most accurate technology transfer indicators?
So the best methodology would be is FMEA. You have to identify your failure modes with respect to your process, product, and the equipment. And then based on that understanding of your risks that is being anticipated and all, we can reduce the most of the challenges in the technology transfer.
Thank you, Mr. Praveen. Another question is from Santosh Kumar from Geetam. What is the importance of half normal plots? This question is, what is the importance of half normal plots?
Yes. So half normal plots will, just a minute, I will just open that so that you'll have a better understanding. See, in a half normal plot, you have the variables of your process variables. Suppose here we are considering two variables A and B, and you have a variable called AB. So the line that is going from the center is an error estimate. The moment the variable that is far away from this error estimate is going to create a more impact on your on your final quality. So this variable needs to be studied thoroughly with respect, or this variable needs to be understood better with respect to your interaction plots or with respect to your control plots. Once you have that control plots, for example, you have a control plot here for this blend uniformity. As I said, the interaction of A and B, which is being determined in the half normal plot, and mutual interaction of A and B only will create you a problem. Not at 30%, if you take it and go at 105, will not have a variation. It's an interaction of both the variations that gives you a indication that my process is more robust in this variation. That will be identified in the normal plot, or what is the variable that we need to study in depth will be given in the half normal plot, will be assessed in the half normal plot.
Mr. Santosh is asking another one question. Is there any software to plot a Haeckel plot? Is there any software to plot the Haeckel plots?
I think as of now, we don't have a software for Haeckel plots. It's like the compression equipment itself. Some of the manufacturing were providing the plots that they are monitoring the compression pressures and all. It will be a printout for with respect to some of the manufacturers.
Thank you. And another last question from my side, basing on my interest, I'm asking. So you are talking about FMEA analysis,
Yes.
where sometimes in the design qualification, we assess the instrument, we go for risk assessment, everything. But
Yes.
in most of the cases, companies cannot afford for a new equipment or modification of equipment. In those circumstances, how you are going to do the process? So are
Yes.
you going to adjust with the existing process, or you will try to convince your management, or go for new equipment or something?
No, that's where we all are in a like we are misinterpreting. From my side, I would say that we are misinterpreting the FMEA, because see, FMEA, once you identify a risk with respect to the equipment variable, it doesn't talk on do replacements and all. Suppose you consider an equipment here in the fluid bed dryer, that the slide I am sharing you. So in that, you need to identify the various variables here. Design variable, design similarities, air distributions, pressure drops, shaker mechanisms. All these things needs to be evaluated at the lab scale, and you need to evaluate these parameters on your commercial scale equipment as well, because you know what you are going to have it on the commercial scale. You are very clear that these are the equipments I am going to have it on the commercial scale before you are designing your process. By looking at a correlation between the various individual equipment parameters, you can fix certain parameters and you can propose an operating ranges on the commercial scale for certain parameters. Those operating ranges needs to be in depth. You need to study in depth in your development during your product development, and understand where I am going wrong, where which process variable is creating more variability. So for that reason, I said that the calculation of air velocity on your commercial scale and the calculation of air velocity on your lab scale will give you a definite correlation with respect to your batch size. Suppose in the air velocity which I am using at the lab scale is 20 air flow, 20 CFM for a batch size of around 100 grams or 200 grams of material. How is that air is going to distribute, or how is that air is going to calculate? So that calculation needs to be done at your lab scale, and that value what you are getting on the air velocity, at what is that air velocity that you are going to achieve it in your commercial scale. You have to determine the air velocity on a plain equipment, and then that air velocity needs to be correlated with the batch size. So there are a lot of statistical tools that you are going to put in place in your development program itself to identify what is critical, what is not critical, and which gives me a better process. Hello? Hello?
Hello.
Hello?
Yes, sir, we can hear you, sir.
Yeah, I think any more questions from anyone? So, are there any more questions from anyone, or just we can proceed further, or just I can disconnect? ప్రసాద్ గారు డిస్కనెక్ట్ అవ్వచ్చా?
ఒక్క నిమిషం సార్, అది పవర్ పోయి జస్ట్ థ్యాంక్స్ చెప్తాను మామూలుగా.
ఓకే ఓకే
ఒక్క నిమిషం సార్, అది మళ్ళీ అది పవర్ పోయేసరికి చాలా పవర్ లేదు, ఇప్పుడు పవర్ వచ్చింది, మళ్ళీ అది ఛార్జింగ్ పెట్టాను సార్.
సరే సరే.
అది అంతా లైవ్లోనే ఉంది, యూట్యూబ్ అంతా మొత్తం. ఓకే సార్, మీరు మాట్లాడండి.
Sir Praveen garu? Sir Praveen garu, can you hear me?
Yes అండి, చెప్పండి.
Thank you, sir. Thank you. Actually, there was a small disturbance because of power cut at our location. We thank you very much on behalf of management, principal sir, and staff of Vigyan Pharmacy College. We thank Praveen Kumar for giving an excellent lecture on the technology transfer issues, and thank you very much. Thank you very much, sir.
Thank you.
సో నెక్స్ట్ స్పీకర్ కనెక్ట్ చేయండి. రెండు చెక్ చేసుకుని పైకి వెళ్ళిపోతాను.
ముందు పెడతాం.
ఓకే, మ్యూట్ చేయండి చాలు, మాట్లాడండి.
Hello, Dr. Ajay Vairale. Can you hear me, sir?
Yeah, I can
thank
hear.
you. Thank you. Thank you. So now we welcome Dr. Ajay Vairale for the next session. Dr. Ajay Vairale, the profile. He done his PhD from JNTU, Hyderabad, and MSc and BSc from Indore DAVV College. And he has a vast experience of more than 15 years. And he worked in various capacities in various multinational companies in Glenmark R&D Center, Mumbai, Dr. Reddy's, Novartis Healthcare, and presently he is heading the analytical research and development at the Center Drug Research Solutions. Now we welcome Dr. Ajay Vairale to give his talk on analytical method development and troubleshooting of HPLC. Thank you, sir. Thank you for your valuable spending valuable time with us.
Thanks, sir. So, shall I share my screen? Can you
Yes,
allow
sir. You
me?
can share your screen.
Okay, just 1 minute.
ఆయన సేవ్ చేస్తాం.
అలా సేవ్ చేస్తే రాదు, ఓపెన్ చేసి పెట్టుకోవాలి. ఒకటే మినిట్ సార్.
చేస్తున్నాడు. Okay, sir, it is visible now.
It is visible. Just 1 minute. Yeah. So good morning to all the panel and the participant here. So I think already Vishwanath sir has introduced me very well. So today's topic is on the analytical method development on HPLC, and this is one of the key instrument in pharmaceutical industry. And we'll go through it, how the troubleshootings of HPLC can be done. So you can see the picture, the father of chromatography. Who invented the chromatography is Michael Sweet from Switzerland, and the first chromatography started with the separation of plant pigments. And in 1906, he started with plant pigment segments. Now, looking at this slide, you can see the transition of such a big HPLC in 1967 to current HPLC. I can't say it's current because again a decade has been passed. There's a lot of advancement has been happened in HPLC, and the first commercialization happens from Waters. This is the slide where it presents very specific on the particle size. Initially, in the column chromatography, in the synthesis, it used more than 100 macrons, but in chromatography, the first is 10 macron. And now today we have a particle size equivalent to 1.7 micron. Now today's topic will go through the basics of HPLC, and for any experiment, we should have a goal setting, what we want to achieve from this experiment. So your target should be number of analytics, how many components you want to analyze, followed by what is the matrix, what is your matrix, means formulation variables, are you going to use a capsule or a tablet or a suspension. Then the third point come on the sensitivity part of the system. Sensitivity means what lower levels you want to quantitate, either you want to do RS or SA methodology. The fourth point comes on the runtime. Runtime exactly is a key factor. How many numbers of samples that will evaluate to decide the runtime. Is your method is a qualitative test or quantitative test? Are you looking for a limit test method or quantitation of impurity at a ppm level? The next point comes what equipment you are equipped with, the make and model of HPLC, the detection levels or the dynamic range of the detector, and what is the skill set is available in your lab. Means how much your people are trained on the concept of HPLC and other things. And is your method going to be reproducible? It's not a one day by luck we'll have the chromatograph or the peaks are separated, and the next day we are facing a challenge. And the last point which comes of the sample preparation requirement. What sort of samples you want to extract? Are you going to use a SP, solid phase extraction cartridges, or a liquid extractions, or the number of methods are available? So these are the key goals which we have to sort before going ahead for any HPLC method. Is I am audible? Hello? Hello.
Sir, we can hear you, sir.
Okay, okay. Because there is no response, I thought.
No, no, no. We don't want to disturb you during your presentation. That's it.
Okay, no,
You
that's
can keep
fine.
your slide in slide mode, sir. Means PowerPoint mode.
Yeah,
Okay, sir.
it's fine. So this is a schematic diagram of a HPLC system, which consists of your mobile phase chambers, then followed by a pump. Then we have a mixer where the component of two mobile phase composition mix. Then injection is there, that which load the sample on the column. And you have seen the heart of HPLC, which is column here. And we have a detector where the based on the methodology, you will have a RI detector, PD detector, UV, and followed by the recorder, which record all the responses which is happening at detector. I didn't know whether this YouTube will work or not. Once I click on it, it will give more clarity on how the HPLC work. Okay, there is some. Hello, maybe this.
Sir, your voice is problem, I think, sir.
Just one.
If YouTube is not working, you can go ahead with your presentation.
Yeah, that I'm thinking.
Yeah.
Dear participants, there might be some challenge with YouTube. So maybe these are easily accessible on net, and you can go through in depth on the HPLC animation. So we'll continue with our presentations, and we'll go to the next slide. What is troubleshooting? So troubleshooting means identification of the problem. So the first we should know what is our problem statement. Then second is the root cause. What is the probable root cause for this problem, and how we can isolate and exactly cause of the problem can be identified. Then how we can rectify this problem and bring back the operations to the routine use. Just an example, if you are getting a peak distorted and you have done a troubleshooting and identifying where is the root cause exist, and once you rectify it, then the routine operations comes in. It means you can start your analysis. So our troubleshooting today with respect to the basic component of HPLC will be divided into 5 sections, which is mostly the mobile phase, the pump, the injector, detector, and followed by the column. So will give the practical experience what we are facing in the lab day to day, and that is the key factor for these presentations. So it may not be available on NAT, but these are the practical experiences we identified as a lab chemist throughout these 10 years. Now coming to the mobile phase, so you can see that the first part of the mobile is that you have to sonicate and decay the mobile phase. If you won't do this, then there will be a lot of challenges. Your pump pressure will be lost. There will be air entrapment in the tubings and lot. You have to clean your inlet slits of the mobile phase tubings. What best you can do, you can sonicate before attaching it to the tubing in a methanol or water, and then attach to the inlet slit. You have to use HPLC grade chemicals and solvent. Why specific I am telling you? Because in lab there are AR grade solvents and chemicals are available, which by the mistake of the chemist or not defined particular standard operating procedure, they can use a substandard grade of chemicals, and that can lead to a number of challenges. What sort of solvent we have to use? So in each solvent, you'll find a UV cutoff value is provided. What is UV cutoff? It's the specific wavelength below above. There is a lot of interference you can see. So you can see in methanol and acetonitrile are the two component in most of the HPLC where the UV cutoff is around 210. So above 210, below 210, if you find there is a lot of interference. Above 210, they won't be give any specific interference. Now mixing of mobile phase. You find that simply people are writing in the methods or in the literature 40-60 ratio or 60-40 they will write. But how to prepare the mobile phase in exactly? Suppose if you are taking a measuring cylinder, taking 400 ml of water and 600 ml of methanol. If you are not writing what is the way you have to prepare it, the analyst can mix in a variable ways. He can take the same cylinder, he will add first 400 ml of water and followed by methanol. There is a mixing of this solvent which generate heat, and the exact ratio, there is a hypothermic reaction will happen, and you will find there is an expansion of the solvent system. So what accurately we are measuring, it won't be, and that will lead to a lot of RT variation. pH adjustment is also a crucial role of mobile phase. Always try to have a separate pH meter for the mobile phase preparation. Don't bring a single pH meter throughout the lab for multiple purposes. That will challenge your pH adjustment of the mobile phase. If you are finding your mobile phase contaminated, please do not use it. Now the reservoirs and contaminate phase. In most of the labs, we use normal detergent, and specific reagents are also used for glassware washing. So we have to very precisely think that it's very cleaned or not. Improper mobile phase or the composition can lead to the buffer precipitations. So we have to take care of that also. And in HPLC, you will find there are a lot of drift issues because of the purity, which I think we have just discussed in the above points that we have to take care during the mobile phase preparation. Going to the next slide, you'll find mobile phase. There's a lot of challenges. You find that one day when you're using the same mobile phase on the same sample, there is no RT variations. But if we are extending your analysis to 24 hours, you'll find that the late runs, the RT has changed of the standard or even the sample. Why it is happening? Because if you have seen that in the practical lab, in the daytime, there are some ACs, window ACs are there. So while leaving the company, the people will switch off this AC to have the power consumptions reduction and all that. And the next and the mobile phase will equilibrate to the next other temperature during its operation, and you'll find a lot of challenges in RT retention time. So we have to take care that we should have a centralized AC, or the temperature of the mobile phase should not be vary during the 24-hour operations. Now the second point is coming on the mobile phase is isocratic mode or a gradient mode. So there is a lot of challenges when you use in isocratic mode. The mixing should be done manually, and I highlighted that the ratio what we are stating 60 versus 40 or 40 versus 60 and how we have to prepare. It should be consistent way. It should not vary with respect to the analyst how is mixing. And gradient mode, that will lead to the pumps, which is not in our hand because there are two compositions in A and B or C, D, and it's up to the instrument. If it's not working properly, we'll have again a lot of retention challenges. Always use fresh mobile phases, and we have to equilibrate the HPLC system. Either we are working in isocratic mode or in gradient mode. As per the practical experience, even for isocratic mode, the choice of mobile phase, minimum we should equilibrate the column for 30 minutes. If you are using a gradient system, kindly equilibrate the gradient cycle at least 3 times before starting your sample analysis or standard injections. You will find if you are not equilibrating in gradient for 3 cycles, the drift in the baseline of the gradient will reduce with each injections. So the suggestion is that always have a 3 cycle equilibrations before injecting a standard. So you will pass your system suitability and there won't be any RSD challenges. So that is the first part of the troubleshooting on the mobile phase. Now the second component is pump. And pump is a very crucial role for this HPLC. And pump challenges or the root cause, if you are finding, it's easy to identify related to pump, which you can easily find through that you have erratic retention time. Means once you start your system, you'll find the retention time is 5 minutes, then it will be followed by 6 minutes, 7 minutes, or erratic. Sometimes it's coming 7, then coming 3, then coming 4. And you find a multiple spikes also, or noisy baseline will appear because it will give a pressure dumping which will be observed in the detector. We have to always prime the pumps, and the sequence of priming our all pumps should be, we should first with a lukewarm water, we should prime the system, followed by isopropyl alcohol and then methanol. And then we have to go in the same way after methanol, directly don't jump to water, again go to IPA, which is miscible in organic as well as non-aqueous systems. Seals of the pumps are very crucial. If there is any leakage, it will be highlighted in non-performance of pump. And if you are using a lot of buffer mobile phase, you will find there is a salt deposition at these seals. So always we have to prime and clean our system on periodical basis. So always, if the seal is gone, always request, we should not deal with this changing of seals and all that. Request, if you have an instrumentation engineer, request him to rectify that one once it is identified. Coming to the next point on the pump, I think first point more or less we have discussed. If you keep your instrument properly, having a preventive maintenance and taking care of all the parts, there will be less chances you will challenge with the pump system. If your piston is broken, replace piston. And one of the major challenges is air trapped in the pump head. So if you have advanced system, they will come with the degassing solvent. So it will degas all the air entrapped in the mobile phase, and you won't face challenges. But in the old system, this degasser is not there, and there's a lot of challenge in the early eras where we have to face this challenge. When we are preparing a mobile phase or selecting a mobile phase, also we have to check the viscosity of those solvent. During mixing of the solvent, the viscosity will change and pressure cycling will be different if there is not a proper mixing. There is a pressure inducer in the HPLC system. If it's not working problem, you will not get the identified the problem that pump is not working proper. So I can say it's a monitor which will give the ECG of your HPLC pump. So pressure inducer always periodically we have to check whether it is working properly or not. Coming to the third slide on the pump, you'll find there are two types of mixing of solvent. There is a high pressure and low pressure models are available. In older time, most of the systems are low pressure model, where the mixing is happening at a mixer from both the channels. The pump will bring from solvent A and B, mix and injected into the HPLC. And because of that, the dwell volume will be 0.5 to 3 ml in the older systems. But now with the advancement in the system, there are quaternary pumps we can use, but it's a low pressure gradient in which all the four pumps will, based on the gradient, they will pump the solvent. It will be mixed in the mixture and then again pumped with the injector to the column. And there is a dead volume of 2 to 5. In high pressure, it will be 0.5 to 3 ml. And low pressure, it will be 2 to 5. So there is a challenge when you do the tech transfer of your method. So what make of HPLC we are using, whether it's a high pressure mixing system or a low pressure mixing system, we have to check it. And if you know that if the method is having a challenge with respect to dwell volume, the method development we should take care that initial 2 to 3 minutes we should make it isocratic. This will resolve your issue of changing the retention time when you do the tech transfer to other sites. So the third component is injector, and you can see that injector is a crucial part because it has to load the sample on the column. If any irregularity from the injector is happen, it will create a lot of challenges. It will give a distorted peak. The reproducibility of injection will be lost, and the system suitability as per the methodology we can't achieve, and we can't run any analysis. Now on the highlighted point number 3, use a pre-column filter. I think we can discuss the guard column in the upcoming sections when we discuss on the column, that how this pre-column fitting will help in resolving the physical degradation of injector seals and contaminations. Now the point number 1, which is highlighting in this slide, where you find that the peak heights, split peaks and broad peaks, we can never imagine that the sample loop, if it's not properly filled, this will lead to this. So it's depend upon the system. Nowadays, there are very precise systems, so this challenge won't be posed. But in the older days, in the 90s area and 2000 decade, we have seen that the companies do not have the good system, or we are using a Riodyne system, where the sample loop is totally depend on subjective to the analyst who is using periodically and the handset of the analyst. But nowadays this is gone case because we have very precise system, auto injectors are there, and these challenges can be moved out. But if any lab is having still using Riodyne, I won't think somebody is using now. But if they are using, the first point has to kept in mind that the sample loop has to be completely filled. It should not be, and that will come with the expertise or the routine experience in the lab. The second point is a part which from the development of the method which comes up. But if that during the development, if it is not taken care, the compatibility of the dialment system which we use for the sample preparation versus the mobile phase, if there is a huge difference between the polarity of your mobile phase and injection solvent, you will not get a symmetrical Gaussian peak. You will get a distorted peak, and the baseline drifts will be always be challenging. I will give a simple example here. If you mix THF in your sample preparations and you are using a mobile phase with more than 60 or 70% of either of methanol or ACN, and your sample preparation is completely in THF, you will end up in a lot of challenge. First, the reproducibility of the peak will be question mark, and the peak shapes will always be distorted. Either the fronting will come or peak split M shape you will get. Okay, so always when we are developing the method, think of what is our dialment system is compatible with the mobile phase. So that is a part of development, but if it missed during development, the method will carry throughout its life cycle. Injection of solvent lower eluting strength than the mobile phase. The same point which I had highlighted or discussed just before, that has to be taken care. Coming to the fourth point, with the advanced system, there is a syringe washing solutions. A separate line is given in the advanced system. So again, we have to take care of that. If our sample or the syringe washing solvent or the priming solvent, what we're using for the injection needle or other component, if that having a different polarity, you will end up in again a challenge with respect to mobile phase compatibility. So always use a weaker solvent in terms of polarity with respect to mobile phase, so you won't end up in these challenges. Coming to the third slide on injector, your peak shape of your chromatography is dependent upon injection volume. So during development, maybe easily you can cross check that with the variation of 10 macroliter, 20 macroliter, 50 macroliter, how the peak shapes are coming. And it will also depend on the previous point which we had highlighted, that the diluent polarity with respect to mobile phase. So even you find that with 10 macroliter, even that polarity is different, you won't face those challenges. But as you're enhancing your injection volume and the mobile phase remains same, then you will get a challenge distorted in the peak shape as long as you are enhancing your injection volume. So injection volume versus polarity versus mobile phase polarity, we have to be very crucial or when we're doing the method development. Same point comes to the injection volume, that resolution I think everybody is aware that is the separation between two closed pair, and it's also depend upon injection volume. If you find that with 10 macroliter you will get a good resolution, but with 20 and followed by 50 and 100 macro, your resolution is lost. So it's not that something column is playing a role or the system is playing a role. It's the polarity of the diluent system and the injection volume is playing a role. So we have to optimize injection volume and the ratios of mobile diluent system, what we are supposed to inject. Coming to the injector capacity, suppose if your system is equipped with a 200 macroliter syringe or 100 macroliter syringe, always use 70 to 80% of its capacity. Never try to go to 100%, because if that 100% if you are using suppose 200 macroliter, you will face a challenge in the linearity. If you're determining linearity of any method or during calibration, it won't pass. So there is a base guideline that you have to use 70 to 80% of injection volume capacity. Means if you're using 200, then minimum you can go with the 150 or 160 macroliter instead of directly going to 200. The higher end, it will always be a challenge. Now carryover problem means that the previous injections what you have injected is coming in the next injection, or after your peak of sample you have injected and you are finding in the next injection also there is a peak which is appearing, that is called as carryover. So with the advanced instrument it was taken care, but rinsing solvent or wash solvent what we are using in the advanced system is also a crucial one. So when we are using a vast solvent and is having a different polarity with respect to the chromatography, what we are using will end up in the carryover problem. Now, again coming to the injection, we'll think injection is having a problem, but it's very surprised that HPLC vials, which is given by the vendor, there is a recommendation that how much you have to fill. If HPLC vial you are filling to the uh, complete filled vial and you keep on injecting, there is a pressure built up because of the complete fill of the vial and it will result in the injector abnormalities. So that we have to take care. The other point is that vial septa, if the diluent polarities, what we are using in the HPLC and the mobile phase are different, and if the polarity of the diluent which we are using for sample preparation is that much crucial that it can extract the some leachables from your vial septa. So we have to very vigilant that what septas we are using and how much is compatible with our diluent system or the mobile phase. If that is identified, I think we can resolve the ghost peak appearance. And these are the practical problems which comes in day to day in the analytical lab, where the without much focusing on these challenges, people go ahead with the development, and later on we find, or even during development, we have seen that there is no challenge, but when it comes to the practical operations in the lab, and the vendor has changed or the supplier has changed, you will end up in these ghost peaks. The last point on the injector, which you said, the viscosity of solvent. So whatever the solvent or the diluent which we are using, its viscosity play a crucial role, as the injector performance is involved a factor of viscosity of solvent. If you see the UPLC systems, it was clear-cut indicated that we can use a particular viscosity solvent because the system itself with very narrow tubings, it was indicated that you can't use a higher viscous solvent. Even the IP, if you want to use as a pure in UPLC, which is having a high viscosity with compared to methyl and ACN, will end up in few challenging the injections reproducibility. So before proceeding for detector, I hope that everybody is going to align with me what I am speaking. If any specific challenges, can raise your flags. Either I have to go very slow or it's optimum for the participant. Hello.
Hello, sir?
Yeah. Can you hear me, Vishwanath sir?
Yeah, Vishnu, hear me, sir.
Okay, okay. So,
Because there is a pause from your side, so we got doubt.
okay.
You can
No,
continue, sir.
okay. No, just I'm requesting participant is the fine the flow we are going, or I have to be a little bit slow.
Yeah, you can continue, sir. No problem.
Okay.
There are some questions I'm getting.
Okay.
We can take over the last.
Sure, sure, sure. Okay, will do. So, dear participant, the next component which we would like to discuss for troubleshooting is detector.
Sir,
So detector, you can use multiple detector in HPLC. You can use UV detector. You can use photodiode array detector. You can use RI. But the overall what I'm presenting here is with respect to generalized one, not specific to detector. There may be specific troubleshootings with respect to RI or ELSD, which can be dealt separately. So first problem which comes from detector is you'll find a leak in the tubing which is connected to the detector, or a air bubble is entrapped in the detector, or there is a cell contamination. So first point including leaks, sorry. So leaks means that the connection problem. Air bubbles, how the air bubbles come to the detector. I think in the first slide when I'm discussing on the mobile phase. So if the degassing of the mobile phase is not done properly, so slowly these minor air bubbles will go through the tubings, and detector is the only part where it can get accumulated after the pump. So you will find that initially you will never face any challenges in the baseline, but you find the next day your runs are lost. You will find there is a very zigzag baseline is appearing. Even you can't identify your peak of interest. So it's because of the air bubbles entrapped. And how you can remove air bubbles? Simply what you can do, you disconnect the column, attach a union, and prime the pump first with water. The first I told you in the pump how you can prime the sequence first with water, then with IP, then methanol, and the vice versa. And the same you can connect through a union to the detector. And there's a recommendation in the detector part that how much flow you can apply. A pressure is limited there, that it should not exceed this pressure. So please take care of that. If somebody is directly starting with 10 ml flow, it will create a major problem. Your cell can be destroyed. So better you should start with 0.5 ml flow, then enhance with 1 ml, and the maximum I can recommend is 2.5. Never exceed about 3 ml for any detector. If you have a ear bubble entrapment, flush the detector for almost 5 to 10 minutes, and then again reconnect and equilibrate your column and start the analysis. That can resolve. Cell contamination. What do you mean by cell contaminations? If you are analyzing this method in which the component is soluble in a particular solvent, and that moment it's passing through the your system, there is no challenge. But what happens in the laboratories, what we will do when we start washing the system, we never thought of that the sample what we are analyzing is dissolved in this washing solvent or not, and we directly start washing the system. And because of the incompatibility or insolubility of this component in this particular wash solvent, it will contaminate the whole system, not only detector, but your tubings, your pump, your injector seal, everything will be contaminated. And when you started the new analysis with other molecule with that mobile phase, and if that is compatible with those earlier used sample, it will elute, and you will find a lot of challenge, additional peaks, ghost peaks, and the whole day your detector will not be identifying the root cause. So whenever we are using any method, when you are using a wash solvent or the wash method, please take care of this. Second problem with the detector, you said there is a lot of spikes comes in the base noise because of the low sensitivity. Refractive index detectors are sensitive to pressure. So we knew that when we are using refractive index detector, is a bulk property detector. It's not related to any chemistry of molecule or any absorption of any wavelength. It's simply a bulk property. It's the deflection of a particular detector from the baseline, which detector will monitor and will quantitate the sample. Always use low volume detector cell. And nowadays, in the previous time, it was there is a huge volume. Huge volume means in maculator, but it's more. But nowadays, with the new advanced system, the detector cell volume is very low and very precise. You can estimate your component. The next point is that different detector requires a different stabilization time. So you can see that the methods which we are used for RA detector, it requires a more equilibration than your UV detection. So as I stated, it's a bulk property detector. So the cell or the whole system get equilibrated, it takes time on RI compared to that of UV. If you go to ELSD, electron light scattering detection, it will take more time than even RI detector. So we have to be very precise when we are selecting the mode of detection. So we should not put a thumb rule for all detector that 30 minutes equilibration is enough for this detector, or half an hour is enough, or 1 hour is enough for that. Based on your methodology, you have to estimate how much equilibration time it has to be required. So that will make ourselves comfortable with respect to different issues in the HPLC. Coming to the lamp energy and reference energy, it should be within the specification which the vendor has provided. Access buffers. So access buffers means, if you see that when we are doing a method development, the combination of buffer versus organic solvents we have to evaluate. If you see that when we are using isocratic versus gradient method, we'll find a lot of challenges in gradient method, that after a certain number of injections, there is a high pressure built up. And what is the reason behind that? Why suddenly after two set of sample analysis, when you put again 16th or 20 samples, there is a pressure build up? Because when we are developing the gradient method, we have to check what is the maximum ratio of methanol or the organic solvent versus buffer. Suppose the gradient is ending up with 10% of buffer and 90% of any organic solvent. Have a practical check in the lab, the physical mixture. You will find that most of the time the problem can be solved if you do a physical mixture check in the lab, and you will find that a higher organic concentration, the buffers are getting precipitated, and we can't see in the system. But if you do this cross check during your development, it will be quite easy to uh, uh, face these challenges of detector sensitivity. For the non-aqueous system, I had not a big list here, but only one suggestion, that whenever we moving from aqueous to non-aqueous, we should have a combination of 50-50% of THF water to clean up the system whenever switching from aqueous to non-aqueous. Now the last point is very surprising, that we have to isolate our detector from cyclic equipment or electronic motor high voltage. This is a practical challenge which we have faced in one of the lab, that we brought 4 or 5 new HPLCs, we established it, and there's a lot of challenge during qualification of this equipment. And what we call the vendors also, they think our instrument is okay, but once we start the calibration, there's a lot of then the investigation is extended to the next level, to the engineering department, and then we'll find that behind the lab, the wall of analytical lab, there is a huge high voltage lines are kept there. And because of the high voltage and the high currents are passing through, there is this the magnetic field what they are creating is giving challenge to this instrument. So whatever the environment we are keeping our detector is also very important. And so it's not that practical challenges is giving I'm giving a more importance to practical challenge than the theoretical which you read in the books, because for such things nobody will write that. Hope you agree on that. Coming to the next slide. Uh, so the only point which I would like to bring as a troubleshooting, that whatever the lamp we are using for detector, it should be of good quality. Means what is the recommended from the uh, vendor. So OEM reflect for the what is the recommended from the vendor. Exact form I will bring it back to you maybe in the end of the slides. But it reflecting the vendor, what is the vendor specification is providing for a particular detector, we should bring that only lamp. If you use a generic source, means in the market you'll find number of companies or small vendors are providing lamp. Ask for a certificate of authenticity of that. It's not meeting the compliance also. If you are using such a generic source and if it's not providing a certificate or a test, it's just simply providing you a lamp that please carry on this lamp. It will work for 2 months or 3. Please, I request not to use those lamps. The slide represents the same thing. If you go to the arrow on the first slide, the upper slide, you will find 4 arrows where the detections, you can see easily you can detect the peaks of interest. While in the below slide where we are using a generic source, the sensitivity of the method is lost. So if you are using a stability analysis, so the trending will be lost. When you compile the data, you will find that each month wise, or you will find the responses, the levels of impurities are showing different, and we can't justify the formulation at that moment. So better to use a vendor specific lamp or the parts what they are providing, not to go with the generic one. If generic is there, please evaluate the authenticity of the generic vendor certificate and their calibrations and all that. Again, this slide presents the same thing which before we have discussed. Here is more precisely it was highlighted with respect to signal to noise ratio. When you are using a very good lamp, which is recommended by the vendor, and we are considering the good detector, you will find the peak of interest will have a good sensitivity in the method. While when you are going with the generic source, first thing is your baseline is disturbed, followed by the loss of sensitivity.
Sir? Hello?
Yeah.
Sir, your voice is disturbing, sir. Is there any disturbance from your side?
No, no.
Okay, sir. Okay.
Is now clear now? Hello?
Yes, sir, you can continue. You can continue, sir. Sir, your voice is not audible, sir. Dr. Ajay, sir? Dr. Ajay? Dr. Ajay?
Hello? Hello?
Sir, your voice is not audible.
Now it's okay?
Yes, sir.
Okay.
You can share your screen. Yeah.
Yeah. So in this slide, so I will again go to the slide number, this slide, and again discuss on the same point. So detector response time is also crucial for the detection. So if your detector setting is varying during the operation, you'll find a lot of challenges. So if the data acquisition time points are more in detector, you will find a good peak shapes and the sensitivity will be high. If the data point collection is low, then it will reduce the peak identification and the separation will be also less. So if you are comparing HPLC versus UPLC or PDA versus UV detector, so in UV detector, the acquisition points are more compared to PDA, and the upcoming ultra fast LC, the acquisition point for the detector is very high. So you'll find in UPLC is lot of sharp pixels because of the detector acquisition rates are very high in UPLs. So this chromatogram represent how the baseline will get disturbed. I think these points we have discussed in the previous slides. So dirty flow cell, detector lamp failing, pulse from the pump if periodic. I think this point I can explain you. If the pump is not working properly, there is a pressure dumping which will be observed in the baseline. So this chromatogram is not presenting a pressure dumping. It will come like a wavy nature. If you have a pulses issues with the pump, pulse dumping means the pump is not appropriately working. It's not giving consistent flow. It's just like when we are breathing and then we'll take a pause and again we'll breathe. It's not a continuously suction is happening or the output is not going continuously. That way it will come to the detector. But we'll think that the detector is not open. It's not the detector, it's the pump which we have to take care. Then temperature effect on the detector, that had explained overall temperature effect on the system, but very particular to detector also there is a challenge, and the sensitivity very specific to RI detector. If the RI detector, if the temperature is varying in the lab, your retention time and the sensitivity of the method will be challenged. Or the problem may be air bubble is passing through the detector, which I think we have discussed in depth before this slide. So there are now again three chromatograms which reflecting the same concept. Unstabilized system because of the pump or the column or the injector. Then detector flow cell leak. You can easily identify that there is no flow. So, and there is a cell leakage. So you won't find any detection. And the last slide, we can see that there is a lot of noisy or wavy nature will appear in the detection. It means there is some air entrapment in the detector itself is happened. So you can see the first slide, which is in red color. So I'd explain you that this practical challenge which we have faced during our one of the lab operations, that there is electronic noise which is observed, and I told you that the environment where we keep the detector is also important. It's not coming from any solvent or any sample or not from any column leak or air entrapment. It's the environment which will give such a spike. If you are finding in your chromatography at any time very sharp peaks like a pin, then you can easily identify that is because of not the system, nor the detector, nor the your sample what you have prepared. It's because of the environment where you have kept your detectors. So maybe you can discuss with your engineering department, is there any specific voltage or any other vibrations are happening which can lead to such a noisy baselines. And high sensitivity, you can see there is a drift in the system which is showing up. So if you're using a very high sensitive detector, now if you makes are providing very high sensitive detector, which maybe in other make of HPLC you never face such challenges, but particularly in this upcoming HPLCs, you'll find these challenges will be appearing nowadays because the sensitivity has tremendously enhanced for most of the HPLCs. So again, it's more or less the same slides, but you can see the temperature fluctuation, how at a periodic interval it is reflecting. Then electronic spikes we have discussed, and insufficient stabilization, which we seen that I recommended that for isocratic methods, minimum of 30 minutes to 1 hour we should equilibrate, or for gradient, you have to give a 3 cyclic gradient run, blank run to stabilize your systems. I'm taking a 1 minute break and then we'll go ahead with the columns. Yes, participants, I am back now and we can go ahead with the columns. So hopefully the other 4 component, you got the essence, what will be the root cause from each component. So this is one of the blacklisted in troubleshooting, I think, with my practical experience, that whenever facing any challenge, people will directly blame column, that we are not getting a good peak shape or wavy baseline, or the experts has to come in picture here, because once you have such challenges in your chromatography, never blame the column, because you have seen that before 4 point, that's I kept column at the last, because the first intention during troubleshooting comes to the column, that column is not working. Every chemist will come or analyst will come to the head that or to the supervisor that the column is not working, but the real challenge is existing somewhere else. So before taking a call that column is not working, I will suggest all the people to relook in the 4 previous component: the mobile phase, what we have discussed, the injector system, the pump system, and the detector. And then we should close, the last will be your column that we have to see. I'm reversing the whole chain or the order where the people first start with the column and followed by other component. So always look for first 4 other component and then come to the column. So why column will pose a lot of challenges during your routine analysis? So if you are using a basic solutes, the compounds which are basic in nature, there is a recommendations to use triethylamine, and there are a lot of columns are available which are base deactivated. Base deactivated means the similar groups in this columns have been end capped with the trimethylsilane, and those are not free for the interaction with the base molecule. And with the advancement in the technology, if you're using a basic solutes, we can go ahead with the polymeric column. And when we are using polymeric column or the new, I will give an example of equity BH column, is a polymeric column. And there is a recommendation for these columns. So we should not directly approach a routine thought process for a washing or the solvent we want to keep in a solvent where we want to keep this column. It should not be generic. So we have to go through the leaflet provided by the vendor for a particular column, and I recommend that we should store in a particular recommended solvent, and not the choice of ours. Then coming to the chelating solutes, you will find that the A grade of silica, which is having some metallic impurity. This is old silica based material. Previously it was used, but nowadays I think no vendor is providing grade A, it's mostly B. So what happens here, the metallic ions which are left in the silica will chelate with the solute. If you are using a compound which can chelate, it will give a tailing, or it will have very high retention. The peak shape is not good. So in such case, there is a recommendation that you can use EDTA in your mobile phase system, which it can chelate with the metal ion in the silica in the column, and the peak of interest won't interact with the metallic ions. So we can eliminate this challenge. So ethylene diamine tetraacetic acid is the EDTA, which we can use, or any other chelating compound we can use. So when we are doing the method development for particularly chelating solutes, I think these things has to be taken care. Again, if you see that there is a recommendation given by the vendors for the columns that you have to operate in a range of 2 to 8 or 2 to 11. So we have to be very specific that whatever the method we are using, we should not touch these boundaries. Means if the recommendation is 2, you should not go to 2, or if the recommendation is 11, you should not operate at a pH of 11. Always go to a -1. Suppose if you are using a column which is recommending a pH of 2, at least you can bring it to 3 or no, 2 to 11. Then instead of 11, you can use it at 10. But don't touch the boundaries of the recommendations what is column vendor has given. That will end up in the extreme challenges that you will lost your chromatography or the column is collapsed. Phase column, I think we can touch in the guard column sections, which is in the upcoming slide, or the ghost arrestor column. We can discuss on that. Again, temperature. Again, so if you see that in the old system, there won't be any provision for any column temperature measure, but with the new advanced system in Agilent, you will find that we can reach up to a temperature of 80 degree for HPLC. But we have to take care of the vendor recommendations. How is recommending, what temperature range is recommending for our column. So we have to operate within the end range. We should not exceed those range. Otherwise, your column packaging will be lost. On the pH part, I think I had discussed just now. Now coming to the dead volume, I think this point again I will discuss in the upcoming slide how the impact of dead volume comes in picture on the peak shapes with respect to the column. So, and you will find there is a void formation at the head of column. Again, we can discuss in the upcoming slide. There are 2 slides for this particularly. And I think we have discussed on the late eluting previous injections, how it can contaminate the upcoming runs. So I recommended always that now the wash method what we are using for any HPLCs, how it has to be done, what the polarity of the wash solvent should be there. When we are changing from one methodology to other, what precautions we have to take. So I think that has been discussed in the previous slides. If any specific questions, we can discuss at the end of this presentation. Now you can see this slide and you can easily find that the column stationary phase has been completely lost, because whatever the injection you are injecting, its retention is lost and it's coming in the void volume. So all retentions are within 1.5 minutes. So if you're finding such peculiar thing in chromatography, I think you can directly blame to the column that this is lost. But still I will tell you that please go through the remaining 4 components before deciding on column. So I think here is the presentation of dead volume with respect to column, which I just told that upcoming slide will discuss. So in the first, you can see that the frit is not properly fixed here, and the mobile phase is getting stationarized in between the ferrule and the column head. So, and it will lead to a leak. So if a small leak is happening, you will find there's a distortion in your peak shapes, or the resolution is lost, or wavy baseline is coming, or there is a drift. You will find injection to injections. The second slide, the second part also peak shapes problem. You can see that there is an additional void has been created if the ferrule heights are not properly or is not recommended as per the system or as per the column. You will end up in a void which is generated at the head of the column. And again, at this end, your sample component will accumulate, and multiple issues you can see. the last where there is no dead volume, you can see there is uh, no solvent is retained in the head of the column. This is the appropriate way to fix the column with appropriate ferrules. So I will recommend in the lab, I will find that different ferrules are lying there, which will add to the column. So during our fixing of column, we should take care that there should not be any leakage, and it should be appropriate ferrule which we are fixing to the column side. If it's not, please take care and replace the ferrule. I think you will save your time and the analysis will be successful. What we discussed now is the presentation in this slide. So you can see that if we create uh, a wide volume in head of the column, you can see a tailing effect is shown in all the peaks. But if it's properly fixed, you'll get a symmetrical and Gaussian peak. So just imagine, because of a small fixing or the appropriate use of parts, we can resolve a multiple challenge in the HPLC. So at the analyst level, we have to be very vigilant on fixing of all these things. So in this slide, you can see there are two challenges which is highlighted. One is on the microbial growth at the columns, and second is if you are using a sample which is not compatible with the mobile phase system, and then it can get retained in the column. So we should recommend that we should use a strong solvent in that the previous solvent system which we are using in the analysis. We can take a particular strong solvent from that and press the column. But don't directly go to solvent and try to flush. Go through the literature, first read it. Is this compatible with my column packaging? And discuss with the colleagues or your superior before starting any column washings or any trial you want to take on the column. Because once it is lost, you can't make a that damage. It's a irreversible damage to the column. So whenever we are trying any washing of column or using any solvent system or any buffers, have a lot of literature search, read it carefully, and then go ahead with your analysis. Coming to the microbial growth, this is a specific challenge when we are using ammonical buffer or the buffer at a higher pH. You'll find after 1 or 2 days, your mobile phase seems to be hazy. So these observations are very crucial. And if you are using a higher pH or a buffer with higher pK, we recommend that we should not use such mobile phase more than 24 hours, and we should establish a mobile phase stability that beyond certain 24 hours we should not use this. Better to use a new one. And there is a recommendation that we can use sodium azide which will stop the microbial growth, or never use a pure 100% buffer for your any chromatography. Always try to accomplish with 5 or 10% of the organic modifier. Either you are using isocratic or a gradient method. The suggestion will be better to use 10% modifier, so you will have a mobile phase stability more than 5 days, and you can use your this mobile phase for your stability sample analysis, because in stability has a huge load of 100 and 200 samples at particular time points. So I think polymeric columns I discussed with you that what is the vendor recommendation choice of solvent to preserve it, because if you vary this, there is a swelling of this polymeric packaging, and it will leads to the lot of challenges. So I discuss on the temperature of the column compartment. Always try to use the temperature which is equivalent to the given by the manufacturer or recommended from the column vendor. Never exceed that. I think this point also we have discussed that if there is a recommendation of pH 2 and pH 8, never use at the boundaries or extreme of these pHs. Always be -1. If pH 2, go with 2.5 or 3. pH 8, even in this case also I recommend to use at a pH 7, not at a borderline of pH 8.1 or 8.2. You will lose the column. I think the last point, I think we are discussing in depth that how a dwell volume at the column head can make a challenges. So coming to the guard columns, what are guard columns? So from the name itself is defined that it will protect your column. So these are guard columns which are of very shorter length. They are provided by the vendor of the same packaging which you have the main column. And whatever the first exposure with the deterioration of packaging, it will happen here. And the strong solvent or the strong buffers which we are using, it will take care of, and your main column will be protected. Apart from this, it will also help in cleaning up of samples. If you're using a matrix samples with a high load of excipients, this will protect. So your main column will be protected, fritz will be protected, and you can use the main column for a longer time. Guard column can be replaces based on the methodology after 15 set of analysis or 20 set, but your main columns will be protected. You can see that the last chromatogram here, which shows the impact of guard column. So with the use of guard column, you can see that how that the contamination peaks has been removed through the guard column. And the slide which I am presenting now is a ghost buster column. So this is a unique technology. It's recently come, I think I can say from last 1 decade, not previously existing in the chromatography. So here you can see that what is Ghostbuster column. So it's particularly used for mobile phase contaminations to be removed through ghost peak arrestor. Now in this slide you can see where it has been attached. So guard columns will be attached to the HPLC column, but ghost buster column will be attached before injection. So you can see that whatever the contaminations come from the mobile phase has been taken care by the ghost buster column, and a neat and clean mobile phase will be given to your HPLC column. And you can see in the chromatograph without ghost buster column how is the baseline, and with the use of ghost how it has been smoothnized. So this is the upcoming technology, and these columns are provided with the patents. So there are 3 or 4 suppliers in the industry which provide this Ghostbuster column, but the packaging and the other component they are not disclosing. But the phenomena is through adsorption or some ion exchange on the metallic impurities or very specific impurities with respect to solvent they are taking care. So my recommendation is that if you are facing such challenges in your blank, in very specific in gradient analysis, please try to use this Ghostbuster column, which can resolve your issue. And better to use guard column where your sample matrix is highly viscous or the matrix is very high in sample. So better to use guard column, very specific for the matrix of formulation. Maybe I can give an examples of suspensions where we are preparing samples uh, 2 grams or 3 gram in 10 ml, and the sample is very dirty, I can say. To clean up, I think better to use guard column, so your column can be saved. That's from my end. Thank you for listening me very Thanks.
Thank you, sir. Thank you, Dr. Ajay.
Welcome, sir.
So now we have some questions from the participants.
Sure.
I think you can see your question and answer section. One question is there.
Just 1 minute. Okay. So the question is on resolution. May I know who is asking the questions?
It's Patel Vishwa.
Patel Vishwa. Okay, okay. So the resolution is not totally depend upon your mobile phase composition. Definitely, if you're enhancing it depend upon the molecule what you are looking for. And if the buffer composition, what is the basic funda which people use here is that you enhance the buffer concentration, definitely your resolution will, but it may not be resolved. So the resolution is mostly based on the capacity factor of your column, the column chemistry, irrespective of the buffer. So, and multiple factors play a role for the resolution, not only the mobile phase compositions. I told you your injection volume also will play a role. People will never thought of that. And second question is what will be done for to decrease the tailing factor of eluted drug? So again, the question comes to the chemistry of the drug. If the tailing happens mostly with the basic molecules. So if that is happening, then you have to use a buffer with trimethylamine. You can add in your mobile phase. Or I had told you that we can use columns which are end capped and the signal groups are not free, that your drug having a chance to interact with the metallic ion cation, which are available in the silica of that particular packaging.
Thank you, sir. There are some questions from YouTube participants.
Sure.
It is from Silambara Saran from Puducherry. He is asking, is there any possibility of using multiple columns in single HPLC? If possible, how many columns can be used simultaneously for analyzing multiple sample?
Yeah. So correct me if I think he's listening to my voice. If correct me if I got his question correctly. So we can use two columns in series, but not beyond that. And it will be in very rare case. We should not try this because it will give a lot of high back pressures to your system. Your pump has to sustain that back pressure, and the reproducibility will be a question mark. So, and won't recommend this way, but in practical experience, I had seen that we had gone up to two columns, not beyond that.
And next question, sir, from Mamush Gebra Mariam. What are the characteristics of selection of good detector?
So on the detector part, first, what is the intention of our analysis? What are we looking for a assay methodology or a impurity? That what is our goal of the method? What we are looking for? That should be the deciding factor for the detector. In general, I will recommend a detector with a big, huge dynamic range that suppose which can capture the linearity from different levels or the wavelength it can operate and easy to operate. And what is your choice? Because I'm again coming back, even the food industry, if you go, they may can use a simple detector also because the recommendation of the regulations in food is not that much. If you come to pharma, there is a huge difference what detector we are using, and that will also applicable to the bioanalytical as well as the analytical lab. Maybe bioanalytical, they may require a very high sensitivity detector if you want to on the HPLC. While in analytical, it may be one or two fold less also will work for us. But if you are very pertaining to genotoxic impurities or impurity level, the detector sensitivity play a major role. Where the upcoming detectors are very good and we can attend up to a nanogram level of detection also on UV can be possible.
So another question from Patel Vishwa. Next
Yeah, yeah.
question is, if chemographic group is not present in drug, we can done UV and HPLC. So I think his question is,
Yeah, yeah. So if the group is not having any chromophoric groups, which is responsible for UV absorption, still we can do that. Maybe you can use RI detector, or you can go to a derivatization of that particular molecule, which will add the chromophore to that group. And the reaction mechanism we have to consider, it should be psychometric and it should be reproducible. It should not that one time we get and the next time when we are trying or move to other lab, we are not reproducing the methodology. Yeah, it's possible.
thank you, sir. Thank you very much for spending the valuable time. And I request all the participants tomorrow at 10 o'clock again, the final day of our FDP program will be there. And I request all the participants to join tomorrow in YouTube or Zoom. And tomorrow there are two sessions, one from Dr. Ravi, IIT Jodhpur, and other from the Dr. BK Sahu, who is from again Regional Manager, NRDC, Visakhapatnam. And I wish a great day ahead. Thank you, Dr. Ajay, and thank you once again. Thank you, sir.
Thanks. Okay.